Hey everyone, and welcome back to the channel. Today we're diving deep into the world of the unseen, exploring the incredible science of microscopy. Have you ever wondered how we can see things that are way too small for our naked eyes? It all comes down to two key concepts: resolution and magnification. First up, let's talk about resolution. Imagine you're looking at two tiny dots very, very close together. Resolution is the ability of a lens to tell you that there are actually two separate dots, not just one big blob. Your eyes are pretty good, but they have their limits. The best your naked eye can do is resolve to points that are about 0.1 millimeters apart. Anything closer than that, and it all blurs into one. To see anything smaller, we need a little help. And that's where magnification comes in. Magnification is exactly what it sounds like. It's the power of an instrument, like a microscope, to make an object appear much larger than its actual size. Think of it like zooming in with a camera. When you see a number followed by an X on a lens, like 10x, it means that lens can make an object look 10 times bigger. So, a tiny object that's just 1 micrometer in size would appear as 10 micrometers through that lens. This ability to enlarge things is the core of microscopy, the technique we use to view objects that are invisible to us, from the millimeter all the way down to the nanometer scale. So, how small are we talking? Well, most of the cells in your body and in the plants around you are somewhere between 10 and 30 micrometers. That's way smaller than the 0.1 millimeter limit of our eyes. Which is why we didn't even know cells existed until the microscope was invented. It truly opened up a whole new universe for us to explore. Now, let us look at a tool that many of you might have used in a science class: the compound microscope. This is a classic light microscope, and it's called compound because it uses more than one lens to achieve its magnification. There's the ocular lens, which is the eyepiece you look through, and the objective lens, which is the one right above the specimen. The really cool part is that their powers multiply. So, if your ocular lens is 10x and you're using a 40x objective lens, you're not getting 50x magnification. You're getting 10 times 40, which is 40x magnification. The object you're looking at appears 400 times larger than it really is. But even a light microscope has its limits. No matter how much you magnify, the resolution is capped by the properties of light itself. For a standard light microscope, the best resolving power you can get is about 250 nanometers. This means if two things are closer than 250 nanometers, they'll look like a single object. The maximum magnification of a light microscope typically tops out around 400x. This is incredible for seeing bacteria and the general structure of cells. But what if we want to see even smaller? What if we want to see a virus or even a single molecule? For that, we need to bring in the heavy hitter, the electron microscope. This instrument is a total game changer. Instead of using light, it uses a beam of electrons to see the object. Because electrons have a much shorter wavelength than light, they can resolve things that are incredibly close together. The resolving power of an electron microscope is an astonishing 0.2 nanometers. That's over 1,000 times better than a light microscope. And the magnification, it's almost unbelievable. An electron microscope can magnify an object up to 2 million times its original size. 2 million. This is how scientists can visualize the intricate, tiny machinery inside our cells, study the structure of viruses, and even look at individual atoms. So, from the 0.1 millimeter limit of our eyes, we use magnification to enter the world of micrometers with light microscopes, and then blast past that into the nanometer realm with electron microscopes. It's this constant push for better resolution and greater magnification that drives so many discoveries in biology, medicine, and materials science. It's all about making the invisible visible. Thanks so much for watching. I hope this gave you a clearer picture of how we explore the microscopic world. If you found this interesting, don't forget to hit that like button and subscribe for more science content. See you in the next video.